So anyway, so the first thing that I would like to mention is that this is not just my work, but it's the work also of a friend and collaborator, Steve Bogdanovich, and both of us, we have been kind of like working on Hydrilla since the Water Institute funded us to kind of like try to understand, as I mentioned before, the patterns of invasion of Hydrilla in New York State. So what I decided to do is I'm going to give you a fair amount of background so you can sort of like, understand what the impact of the research is, or like where the research that we were funded for falls into place. Okay, great. So I guess that the first thing that I would like to kind of like highlight is that what we call Hydrilla, I mean, like the Latin name Hydrilla Verticillata, is actually kind of like a complex group of plants that they have a wide distribution. They can be found all over the world, and that they have like a lot of morphological variation, and that morphological variation that hints at sort of like differences between different plants or populations that not necessarily actually correlate with the geographic origin of those plants. So you can find a lot of variation, and that doesn't necessarily like give you any information about the origins of those plants. So that high morphological variation is also associated with an extensive amount of genetic variation. And this is not our work. This is like previous work that has been done since the 1980s, 1990s. And one way in which we see this amount of genetic variation is in the way that the genetic material is arranged in chromosomes. And early work showed that most of the populations in the United States that have been karyotyped, they actually have 3 sets of chromosomes, of 8 chromosomes, making a total of 24 chromosomes. And that despite the fact that most of the plants, as I said, are triploid, they have 3 copies of each of the chromosomes, we have found, well, the royal we have found diploid plants. And to some extent, I think that in 1 or 2 instances, we have seen actually tetraploid plants. And all of those can be found, for example, diploid and triploid plants can be found in Washington, D.C., and they have like no correlation, again, with what we call the biotypes of Hydrilla that we find in either New York State or in the Northeast, for that. So it's complex. The genetic information is arranged in complex ways, and that is also showing at a different genetic level, which is when we look at a specific genetic loss. So those are like specific genes that we can actually sort of like look at this level of genetic variation in some of the ways, as I said, and then we can look at different genes. So now what I'm going to give to you is sort of like all the genetic knowledge download that there was before we did this here at Cornell. So in the same way we kind of like, track maternal ancestry in humans, looking at mitochondrial DNA, right? We can do the same in plants, but instead of like looking at mitochondria, we look at chloroplast, that is chloroplast gene. In this case is the gene tRNLF. Doesn't really matter, right? And so there's a previous genetic study, like worldwide study, in which was trying to track the origins of the invasive populations of Hydrilla in North America. And then like the home message here is just that the monoecious Hydrilla is a biotype that is found in the Northeast that is associated with a particular haplotype, chloroplast haplotype, displayed in orange, is different from that of the dioecious plants that are mostly found in more temperate weather, like kind of like the South, but also kind of like in the West Coast. And that was sort of like the extent of the knowledge till 2020, in which actually someone else like looked at some of the populations of Hydrilla in the lower Connecticut River, and what they found is that they're actually very different from any other plant that had been described before, and neither matches the standard monoecious biotype of Hydrilla found for the most part across North America, in the Northeast, or the dioecious types of, as I said, the West and the South. So that was kind of like an idea, and then that kind of like put or pinpoint to slightly different geographic origins for those different biotypes. That's not the whole story, right? We know that, the mitochondrial DNA in humans doesn't tell us the whole story of our ancestry. In a similar way, the chloroplast is not going to tell us the entire story of how monoecious or dioecious hydrilla made it into North America. Now, so these people that did all of these work also look at two other genes. And one of the genes that they look at is one gene that is called ITS. It's an internal transcriber spacer. It's a very famous gene. And it's only famous because, a lot of people use it for different phylogenetic analysis in many different plants. And there's a lot of things that we can talk about here, but perhaps the most important one is this. I mean, like when we look at this particular gene, we find dioecious plants in the US are a single group that is mostly related to plants in Nepal, India, Burundi, and Pakistan. And dioecious plants actually have two sets of genes. I mean, they have one copy. This is a nuclear gene that is more related to the genes that we find in China, South Korea, and Australia, and another copy, another gene that is actually quite different from the previous one, and that is related to a different geographic origin. So this was, among other things, used to claim that the monoecious hydrilla, that is the one that we are expecting to see in New York State, is actually what we call an allopolyploid. That is a polyploid, has more than two copies, in fact, 3 is a triploid, and that perhaps is the result of a hybridization between different lineages that have different geographic origins. So when we look at other genes, and this is the last gene that I'm going to show you in detail, this pattern of sort of like hybrid origin seems to hold, but it does complicate things a little bit more. So when we look at a gene called PDS, that is a phytodesaturase, and I think that now I'm going to tell you why this gene is relevant in a minute. What we find is that actually the structure is slightly different. So now both dioecious plants and monoecious plants in the United States have different copies, at least 2 different copies of these genes. And all the plants that have been genotyped so far, they're always like that. They always have 2 copies, so they're always heterozygous plants. And that's also the case for those monoecious plants that are found in the lower Connecticut River, despite the fact that those alleles are actually quite different from the monoecious alleles that we find in the rest of the US. I mean, hopefully this kind of like makes a little bit of sense. I mean, at the end of the day, is just trying to convey that, yes, a lot of the genetic variation is found in the heterozygous stage. That is complex, and that this complexity is associated perhaps with the fact that these plants in the US are actually allopolyploids, again, kind of like the result of an hybridization event between different lineages with quite different geographic origins, and of course, different genetic patterns. So why am I spending time in all of this, and why am I spending time in this particular gene, is because this particular gene is actually part of the carotenoid biosynthesis pathway, and is actually the target of a herbicide that is one of the most common herbicides used, not just in New York State, but across the country for treating the populations of hydrilla, to control the populations of hydrilla. So this PDS, phytodesaturase, can be kind of like affected by fluoridone, and once you kind of like break down this particular step in the carotenoid biosynthesis, then you start to accumulate intermediate products, and this results in sort of like mortality for the plant. So now we know, and this is what I meant to highlight here in red, is that in populations in Florida, and those populations are always dioecious, we have found mutations somatic mutations that are actually responsible for resistance, herbicide resistance, fluoridone resistance in those populations of Dioecious hydrilla. So this is, believe it or not, the extent of all the genetic knowledge that there was before we started looking at these plants in New York State, as far as I can tell. So what we did, and this is where, you know, the Water Institute comes into play, is we meant to kind of like use high throughput sequencing, like next generation sequencing, to kind of like answer several questions. One is, can we identify the hydrilla? What kind of hydrilla do we have in the state? And the idea is that we have monoecious hydrilla, but it's also possible that there is dioecious hydrilla that has not been morphologically identified. And this may be more of an issue as we believe that the temperatures will keep rising, particularly in the south part of the state. Then can we find evidence of any fluoridone resistance? I mean, this targeting or like looking at this particular gene, we thought that it might have like, you know, a direct impact in some of the management, in some of the management of populations in the state. And for doing that, actually, we look at, or we did what we call targeted genotyping, and we target the chloroplast gene that we have like seen before, and the PDS gene that I just tried to describe. So that was not just the only two questions that we want to answer, but the last one was kind of like trying to determine the connectivity patterns, like trying to find out how the different populations of hydrilla in the state relate with each other, and seeing if we can find like a correlation of that genetic similarity with measures of connectivity, geographic distance, amount of boat traffic that goes between one place and the other, presence of highways, anything that will kind of like help the managers to figure out where to put the effort to stop the secondary spread of hydrilla in the state. And we do that, or we aim to do that, with more of like a complete genome type of approach, like, or a genome-wide sequencing approach, by looking at multiple, I'm talking about like tens of thousands of sites across the genome in the plants that we collect. So how did we do that? I mean, in collaboration with the DEC and the Water Institute, we collected plants in 11 locations across the state that translated into about 104 plants that came into the lab. And that represented like, I mean, of those 104 plants, we're able to extract high quality DNA from 84 of them that are the ones that were part of the subsequent analysis. And then on those samples, we did that targeted genotyping that I tried to describe, and this genome-wide genotyping. So the targeted genotyping, we target those two genes, the chloroplast gene and PDS, and we collect about 4 million DNA reads on those 84 plants. And for the wide genotyping, believe it or not, we collected about 1.3 billion SOR reads of DNA. Of those, most of them actually pass all kinds of filters. It was like a really good data set, which translated in about, I don't know, 170,000 loci across the genome of hydrilla that we can look at. And believe it or not, all of that translated only in about 450 variable sites that we were capable of tracking in at least 95% of the individuals that we genotyped. So very little. I mean, like I was talking to you about extensive genetic variation, but when we look at the populations of New York, actually we start seeing a reduced amount of genetic variation, if that makes sense. So what are our results? Well, so one way to look at the results is to generate what we call an haplotype network. And this haplotype network is similar to the trees that I showed you before, in which like you can see the relatedness of the alleles that we sequence in this network. So like each of the circles will actually represent one of the alleles. And those alleles are connected with other alleles by these little marks. And those little marks are mutations, right? And then the size of the circle tells us something about the abundance of that particular allele in the pool of plants that we have sequenced or in the population, whatever. So here's a network, and this is a network that is kind of like that we produce based on the previous sequencing efforts that they were done with all the studies. And then in here, I identify for you, so like the alleles, the circles, right, that correspond to Dioecious hydrilla in the US. Now, in the next color, like in blue, I identify for you the blue as the monoecious hydrilla. These are the haplotypes that we knew that they existed before. And I'm showing here the two haplotypes of the Connecticut River that are the lower Connecticut River that are like very different from the rest of the US monoecious. And now finally, I'm showing you the results of our genotyping in which New York actually has these two alleles. Again, the plants are always heterozygous, so they always have one allele and the other. And actually, they are indistinguishable from any other plant that has been genotyped in the Northeast, right? Those are sort of like the states where you can find actually these two haplotypes. So yes, we cannot distinguish the plants just based on PDS, just by a single gene. I mean, that was not a surprise. But what actually the technique that we use to genotype these plants allows us to do is to identify not just the two copies of the gene that I was just trying to describe, like copy A and copy B, but we can also kind of like, and we can distinguish those copies based on either like, single nucleotide variants that they have or insertion or deletions that may have in their introns. But we can also actually identify patterns of variation that are consistent with somatic mutations, right? So those somatic mutations, much as like the ones that you will expect in any tumor, they kind of like start accumulating in the tissue of a particular individual plant. And if you were to kind of like take that fragment or take that part of the plant, and if that plant was to reproduce asexually, can kind of like carry that somatic mutation with it. So we're able to kind of like look at those somatic mutations, and therefore we look for evidence of perhaps incipient accumulation of somatic mutations that have to do with fluoridone resistance in the populations of New York State. And basically, we're looking at just one particular mutation that has been well characterized, that is in the codon 304 of the gene PDS, and 3 different mutations have been characterized, and each of them like confer like resistance, like from low to medium to high resistance to the herbicide. And if we were to find any evidence of those, like we were expecting, like these changes that you can see in here. Now, that's kind of like what you will see if those mutations are actually present. But as I see here in this cartoon, basically we found no evidence whatsoever that there are any somatic mutations that seem to start or that are beginning to accumulate in the plants of New York State. For all we can tell, there's nothing to it. Now, I mean, there's evidence of some other somatic mutations, but for all we know, we cannot link them to any resistance to any herbicide, and that's it. So I guess that was sort of like the first 2 answers, right? Or the first answer, like, no, there's no evidence for accumulation of resistance in New York. Now, the other result that is like perhaps less exciting, if you wish, is like, oh, well, so what kind of hydrilla do we have? And I told you that perhaps, there was some dioecious biotypes in New York. The answer is like, we only found a single variant of chloroplast, which actually corresponds with the lineage of monoecious plants. So for all we know, all the evidence tells us that, the plants of New York have some depleted genetic variation. They should be all susceptible to the fluoridone herbicide, and they all belong to the monoecious biotype, which for the most part confirm like what we thought it was the most likely scenario in the study. Now, as for the genome-wide sequencing, Well, it was a bit of an adventure, and it was a bit of an adventure because we have a fair amount of experience in this type of technique, but we have never seen what I'm going to try to describe here. So usually how it works is that you take the genome of each individual plant, you cut it, and then you take those cell fragments, you read them, and then you kind of like map them back in the genome and figure out how much genetic variation each of those fragments carry. And each of those fragments actually, you know, is what we call a locus, right? Plural loci. So if you're with me, now you take the second plant and you do the same, you cut it in the same way. And we do this cutting with restriction enzymes. Also, it's a chemical cut. And you genotype the plant, and then you find, you know, the genetic variants that they have, and you do this with each of the plants individually. So the assumption, right, is that the cuts that you made in plant number 1, plant number 2, all the way to your sample size are, for the most part, very similar. So you always recover information for each of the fragments in each of the plants. Does it make sense? This is like, makes sense to you? So
yes,
what we found, despite of all of that logic, and this is what it holds for most of the studies that we have done, what we were not expecting is to see exactly this. So look at this distribution. This is the number of loci. This is the number of fragments that we look at. This is from, you know, from 0 to up to 50,000. And this is how many of these loci, right, are found in 1 plant, 2 plants, 3 plants, 4, all the way to 8. So remember that we genotype out of like 84 plants, right? So if my logic that I tried to describe was right, most of the fragments that you see, right, should be genotyped in all plants. I mean, like these shouldn't be here, these should be up here. And actually, you should have like a few loci that you only see in 1 particular plant. I mean, like that's kind of like the error that you expect. But the trend is absolutely different. I mean, like this is absolutely breathtaking. I mean, like most of the fragments that we can read, most of the loci that we can read belong to 1 plant and just 1 single plant and no other. Now, why is all of that? I don't have any good reasons, or I have some hypotheses, but I cannot really explain it. I mean, it may be a technical error, but none that we can actually pinpoint to, or perhaps, and maybe this is the closest answer that to perhaps reality is the plants that we get. They are not growing on a clean petri dish. So in other words, they come with a lot of epiphytes, and perhaps the communities of epiphytes that they carry are like very different and produce a lot of noise. And a lot of those sequences that we read are not necessarily coming from the tissue of the plant, but they're coming from all the epiphytes that are growing on that. And that will kind of like throw out, you know, some of the logic or some of the expectations that we have. So anyways, for better or for worse, that's the reality. Like we spend an incredible amount of time and money trying to figure out this, but that's how far we got to the possible explanations. But beyond these, we were able to collect data on about 169 loci, 169 fragments that at the end of the day, we thought they were like clean and informative. And each of those fragments carry several single nucleotide mutations, SNPs, to a total of about 500. So now the results that I'm going to be showing you, they're based on the variation across the state in these 500 nucleotide polymorphisms that we can track. So no more than that. So one tricky part about like working with triploids is that it is possible that recombination in alloploid plants is suppressed. So there's no recombination between the two different set of chromosomes that are coming from two very different genetic backgrounds. And if that is the case, there's a clear violation of, the population genetic principle that is called Hydrilla Verticillata. So that means that we cannot use some of the statistical techniques that we typically use to analyze population structure. And the way we thought about it is, well, maybe we can just use multivariate analysis, because it simply is not affected by all of this. So if we do a classical multivariate analysis, this is the results. There's the results. Seems that Blindenberg, like the plants from Blindenberg, are slightly different. But other than that, I mean, there's like really not a lot of structure. Now, a standard principal component analysis is maybe not the most appropriate multivariate technique to look at this type of data. And there is another technique called DAPC. Stands for discriminatory analysis of principal component analysis. And this one actually does perhaps a better job at distinguishing the populations that we have in New York State. So let me walk you through all of this. So the first signal that we see is like, yes, seems that those plants in Blindenberg that I told you that they seem to be different actually might be different. And so the populations south in Long Island in Ronkonkoma. Now, I mean, like, once you're like looking at this level of variation and like all the rest of the state is like up here with no, all the other plants kind of like overlap with each other. One question is like, okay, well, how different is Blindenberg? What is the real distance? What is the real genetic distance between these plants, the Blindenberg plants, and the rest? So this is what I try to summarize in actually these two axes. So believe it or not, all of this distance that we see here that seems to be like massive, actually is just driven by allele frequency differences in only 8 SNPs. So at the end of the day, most of the distance is that one of the SNPs is an A in all populations, but is a T in Blindenberg. And similar to that, there's one SNP in which all the other populations of New York are a G, and the ones in Blindenberg are a T. So tiny genetic differences. We're talking about 8 genetic differences in I don't know, all across the genome for all we can tell, right? Now, I mean, that distance is even less so in between all the plants of the state here and Ronkonkoma. So that distance is just like based on the fact that there's 2 different SNPs, just 2 single mutations that make a difference in frequency, make a difference between the plants in Ronkonkoma and those of the rest of the state. So the view that does give us is that, well, there's very little genetic variation, right? And yes, perhaps we can point to 8 or 2 different mutations that make some of the plants in Blindenberg different, but perhaps there's a bit of an artifact. And the reason there's a bit of an artifact, or perhaps there's a bit of an artifact, is because this DAPC is actually trying to pull, it's kind of like working with information on the populations, and it's trying to pull those plants from Blindenberg as far apart as possible, mathematically, from anything else. A way to kind of like get around that and to kind of like confirm these results is to kind of like repeat this analysis without giving the computer information about the populations. So if we do that, actually, yes, we still see the cluster of the populations of Blindenberg. So, sorry, the population of Blindenberg. And we see 2 other genetic clusters that are like, as you will expect, very similar to each other. But neither of those 2 clusters that the computer can pick on actually have any sort of like association with geography. So what is our interpretation of these is that actually these two clusters are driven by some small sequencing error and some of the somatic mutations that we talk about. And those somatic mutations actually do not show any geographic association. And if you wish, like at the end of the day, all of these argues that there's a single clone of Hydrilla present all across the state. So if you ask me, I mean, like all of these data, I mean, seem to tell us that there's no evidence whatsoever of sexual reproduction of Hydrilla in the state. There was a question about if actually monoecious Hydrilla or not, sorry, if monoecious Hydrilla or not actually has sexual reproduction in North America. It actually points to the fact that it's a single clone in New York State is just like a single individual plant from a genetic point of view, if you wish, and that plant is most likely spreading through just fragments across different populations. Those fragments can carry some of the somatic mutations that accumulate as a result of lack of recombination in alloploid plants. And because it has not been time enough since the introduction of Hydrilla in the US, those somatic mutations have not increased in frequency enough that actually results in any population structure. So this, or the corollary of this, is that actually there's no population structure and we cannot track the origins of the different plants across the state. So it's not possible to kind of use this genetic information to study connectivity or to inform the managers of how the plants are moving across the state. Beyond that, also, we have no evidence for any of the somatic mutations affecting the performance of fluridone as a control herbicide for this plant. And once I said all of these, perhaps there's still hope for finding a structure or for trying to inform managers about connectivity using genetic methodologies if we look at a different part of the genome, and that part of the genome won't be the SNPs that I tried to describe, but it will be looking at highly repetitive regions of the genome that are highly variable and perhaps contain a little bit more information, or the information of population relatedness that we miss, despite the fact that we're looking at the entire genome. So that's about what I have, and I hope I don't know, I'm keep that sweet spot between being too technical and too naive.
Thank you for that, Jose.
Cool.
Maybe, sorry, or while you get set up, Ashley, I'll just say for folks in the room, Hydrilla is an invasive plant. It grows very densely and it tends to outcompete native plants. Sometimes it can provide cover for fish, which might be a good thing. It can also clog boat launches and make it really difficult to recreate in other cases. So, it's not always bad in all cases, but it is very prolific. And once it starts somewhere, it can be very difficult to manage. So that's why it's near the top of the list for invasive species management in New York. It is definitely in Cayuga Lake in several locations. And it is also been found, I think I'm not saying anything new, in the canal, and obviously in all of those places that Jose showed. So in at least all of those locations, there's Hydrilla. And it's sort of a yearly thing. It pops up in some places, and then we try to manage it and push it back in other places. But so that's why the question is coming up about Cayuga Lake, because it is a very active issue here. And there is active herbicide treatment, plus pulling in some cases. There's talk of there's plans to dredge the inlet. Lots of different types of management going on for different reasons. And how all of those different activities will impact Hydrilla is still a little unclear. Ashley. Thank you.
I'm gonna say very similar things here in a minute. I was like, oh no, he's stealing my entire presentation here.
All right.
No, you're all good. So anyways, I'm Ashley Morris. I'm the Aquatic Invasive Species Coordinator for Region 3, which is the lower and mid Hudson Valley. So, you know, about 3 hours away from you guys that are in Ithaca. And I work in collaboration with the DEC and WRI. Maybe this will go forward. Huzzah. So on the management side of invasive species, the name of the game for us is really to rapidly, you know, detect species as soon as humanly possible once they become introduced, and then to implement rapid and effective treatment. To kind of visualize how that looks, a lot of people have used this invasion curve, showing the introduction all the way there on the left, where it hasn't been in an area for very long and it hasn't taken up a lot of space. As a species becomes established, it will grow and take up more space and become more and more difficult for us as managers to manage. And where that detection takes place also will have an effect on what sorts of management actions we can take. So all the way on the right, you see in the red, it's local control and management only. So it has been there for a long time. It's very established. And so a lot of the times all we can do is clear off a boat launch or make things accessible for people. But as you move toward the left and maybe a species hasn't been there for as long or hasn't taken up as much area, things like eradication, like completely getting rid of the species, becomes a little bit more likely and a little bit easier. So the role of research science and a lot of the things that Jose has been working on with us is making sure that when we take those rapid responses, that they're the most effective responses possible, so that, you know, we can achieve eradication in maybe those green and yellow zones. And then also research can be very helpful in the early detection. So hopefully we can detect a species as soon as possible so we can take those actions. Something else that Jose had touched on that might be sort of a future goal of his work is understanding the connectivity of species. And so for us, that really precipitates out to understanding what the vector of that species is. Hydrilla is a submerged invasive plant, so it grows underneath the water, although when it becomes very prolific in an area, it can top out, meaning it takes up the entire water column. Which you can kind of see in that boat picture there. It's supposed to be a submerged species. It is barely submerged in those locations, but understanding how a plant has gotten there and what is taking it around is also going to direct the types of management that we're going to do, and then also the types of education and outreach that we're going to try to do as well. You know, if a species is spread by boat, we're going to target boaters and make sure that they're cleaning their boats and draining all the water out and not accidentally transporting plants across the state. If we see that there, you know, is not as much connectivity, it could be something like aquarium stores and people buying plants for their aquarium and then dumping their aquarium out, which happens a lot. And then we might, you know, try to target our outreach toward those types of retail establishments, which we've also started an outreach sort of event for. Things get a little bit trickier when the vector is something like Canada geese. You can't convince a Canada goose to please clean its plumage every time it goes from one place to another. But there are other more creative management solutions we have for those. Brian brought up some of the treatment actions that we can take. These are more general. I'm not saying necessarily specifically for any type of plant, but we have chemical treatment. A lot of the things that we look for when we do chemical treatments are what that chemical has impacting on the target species, so the species that we want to get rid of. And that can also be things like resistance for hydrilla. You want to make sure that you're not using a herbicide that could create resistance or that a plant already has resistance to, because again, that sort of eliminates that rapid effective response. You want to understand the non-target species effects. So if there are any native species that could be impacted by chemicals. And then also things that are a little less, you know, connected to scientific research, like cost and permitting. There's also manual treatments, which usually takes form in hand pulling. There are some species that hand pulling is super effective, or at least more effective than in other species, which you also have to take into account. Is this a species that you can hand pull? They tend to have less non-target impacts, but you still want to keep in mind if there are rare plants, you want to make sure that people going out there pulling them know what those rare plants look like so that you don't accidentally pull the wrong thing. This is a really high effort treatment plan. It takes a lot of time and a lot of people to make the amount of difference that you need for effective treatment. And then you need to have safe disposal of those plants so that they don't reinfest that area. Then there's mechanical treatment, which can be something like this harvester, or it can also be something like dredging. It's technically a mechanical treatment. You want to make sure that the species that you're targeting is going to be impacted by that mechanical treatment. If the harvester can't reach your plant, it's not going to be impacted by it. Machines also aren't specific in what they're picking up, so there's some non-target impacts to be considerate of there as well. And then a lot of these machines can also be quite costly, and there's also usually biomass to dispose of the same way that you would have to with hand disposal or hand removal, where you just have to be careful where it goes. And then the last control that we can have, or one of the last main controls that we tend to look at when we look at management is biocontrol. This one's usually impacted by just generally the availability of the biocontrol. There are not many biocontrols that are currently available within the United States. It takes years and years and years of research and funding to find one that isn't going to have impacts on the native species. That amount of research costs a lot of money. And then you also have to keep in mind that now you also have to manage usually an insect or something in addition to managing those invasive species. So that can be a little bit difficult, but it is also something that people tend to do. And I know that Cornell has quite a bit of research into biocontrols. So for hydrilla specifically, it's a little bit of a tricky one. There's all kinds of treatment options for you. Unfortunately, in the United States right now, there's no viable biocontrol. So we can't do that yet, hopefully one day. But you can try a couple of different things. Generally, what has worked best in our region is that herbicide treatment. We found that when we try to hand pull hydrilla, it's difficult because it's under the water instead of on the top. And then with mechanical treatments, you also do run the risk if you pull it at the wrong time of fragmenting it into little pieces, and it can actually potentially make the problem worse. It can be done, but you just have to be mindful of those things. So in my region, we've mainly done herbicide treatment, chemical treatment. But what Jose's research and other people's research can really help us with understanding is what chemicals specifically or combinations of chemicals that we should be using that is going to be most effective on the plant and least effective on the non-target plants. You know, we want to hurt the invasive species. We don't want to hurt the natives as much as we possibly can. A use case for these kinds of treatments and how understanding the genomics of hydrilla has been really helpful is understanding the interaction between two water bodies that are in my region, which is the Croton River and Lake Sebago. So the Croton River had a pretty large infestation of hydrilla. We created a management plan that used fluoridone, which is that herbicide that was mentioned earlier. And it had really good outcomes. We treated the water for a few years, and this year we came back, and that one piece of hydrilla in that sandwich container is the only hydrilla that we found this season, and we were able to hand pull that. And that has been fairly consistent for a couple of years now, that the hydrilla really hasn't grown back after that treatment. We're able to use that information that we don't have a large genetic diversity, and that genetic diversity is not reflective of herbicide resistance, in being able to predict the outcomes of a very similar treatment, unfortunately. In Lake Sebago, there was a hydrilla infestation found. We've created a management plan utilizing fluoridone, and hopefully, based on the information that we have, we'll get very similar outcomes to what we have done in the Croton River. But you know, the interactions between management and research are really important to help us get that early detection and rapid response that we need, especially in a plant that's so difficult like hydrilla. And that's all I have. Maybe.
Thank you, Ashley.